we need some time also for us to adjust. Do you have the, yeah, you have the control now?
No.
Okay.
Okay, so let's do it again. So welcome to my master thesis defense entitled Exploring the Plankton Embryology and How to Bridge the Gap Between Morphology and Taxonomy. As I said, I did my internship in the Institut de la Mer of Villefranche-sur-Mer between the Laboratory of Developmental Biology and Oceanography. Few words about the context of this project. First, what we call plankton is all living organism drifting with the currents. And as you can see on the picture on your right, it gathers a huge biodiversity, crossing all the kingdom of life, coming from the plants, the animals, but also protists and even bacteria and viruses. And these organisms are responsible of essential ecosystem services because they are involved in biogeochemical cycles and they are at the basis of the world marine food web. However, it's also very sensitive organisms due to environmental factors, and particularly during their early life stages when they are more fragile. And in a context of global change, we can guess that that will have some impact, but we are not able to understand which one. Okay. Sorry. So to study the plankton, one of the main tools is traits. Traits are morphological, physiological, phenological features at different scales. So it can be from the cell to the whole organism. And the most important one I will detail right now, and this will be the ones that I will use during this presentation. So there is a size and shape, like this colonial cycle or this microscopic mollusk, but there is also the color and transparency. So morphological traits constitute a way to study inter but also intra-species variability. I've shown you previously some different group of organisms, but here you see only copepods and already many morphology different. And so morphological traits constitute a robust tool to transcend the taxon and the local specificities, because they provide also accurate estimation in terms of physiology, behavior, and life traits. Like, for example, swimming behavior, metabolism, or UV protection, for example. So coming back to the eggs and embryos. Here, I've told you that it's copepods, but also there is bags of eggs on the mother. They are still attached. And we think that already the morphology is an heritage from the parents. And it's also a marker of their investment, for example, in number of eggs, size, but also pigmentation. And this trait will also change according to the development, because the egg is at the beginning one cell state, and it has to become multicellular, but also the embryo will consume the reserve that it has, so you can change many traits. So it reflects clearly the functional trait and the survival of these individuals. And what is particularly important is that it concerns also non-planktonic species. I mean by this that the early life stage is planktonic, but then the adult stage are not. And so in this case, it means that if the larvae are exposed to stressors, maybe the whole species can be in danger too. However, today there is a lack of integrative studies and complete databases regarding the morphology of these early life stages, compared, for example, with huge database like Ecotaxa that we have for the adults. So now we have a problem because there is a missing connection between early life stages between themselves and with also the adults. And we are not able, when we have an image of eggs, to know from which species it belongs. So the question of my internship was how to estimate the diversity of these morphological traits in the wild plankton eggs, but also their variation during the embryonic development. So for that, we have designed several steps. So I will go through most of them. First, how to develop a complete, adaptable, if possible, affordable and easy to use workflow to record these morphological traits and to analyze if there is some possible correlation between those traits, and to monitor temporal variation during the embryonic development. And all this morphological data has to be then connected with the taxonomy in order to make the connection. For today, I won't go right now to this last point because we didn't have all the data in time, but you will see that we could have already some preliminary results. So the first step is to design the workflow and first to collect plankton. So we have did it in the Bay of Reference here. Then we have to sort the eggs. And here it's a beautiful sample, but find the eggs in this soup, you see it's already found needle in a haystack. Then the aim is to maintain the eggs and the embryo in culture and to be able to follow them. And as you can see here, there's some picture from the beginning. So again, here I show an example with a copepod mother with a bag of eggs, changing according to the development state until the hatching and the release of the nuclei here. And in parallel, in order to have a higher resolution in time and space, we have selected few individuals that you were supposed to see developing here. And for that, we use the mounting system to maintain them during the acquisition. The classic mounting system is between cover slip and slide, as you can see here. However, it was not adapted for round eggs. So we created a second mounting system with a 3D printing mold with different size of spike here in order to create some holes in the agarose. So I show you with the drawing again, the mold will create some holes with different size or different diameter. In each hole, I will put some egg, and like that, I could have many acquisition without disturbing the development. And what you can see here is the possibility to add an agarose cover on it in order to avoid the floating eggs to go out of the field of view. And here, again, it was my favorite video, so I'm sorry, but you would have seen the whole development of this bony fish from the early stage of the development until the hatching. So after all this image acquisition, of course, we need image analysis to extract the morphological trait. Yeah, but I don't want to detail you now because it's too long. And the DNA barcoding here to extract DNA sequences to know from which species each individual we have acquired was belonging. So again, I won't detail right now the barcoding because I really want to focus on the first result in terms of morphology. So we acquired the snapshot, as I said, and from that we were able to record the morphological trait. So we use 30 features to measure the color. Color in an image is defined by the hue. So in our case, going from the orange to the blue. The saturation, which is a kind of intensity of the color from very pale and very intense color. And the brightness, so the lightness, coming from very dark in case of transparent sample, but very bright in case of opaque sample. So it was for color and transparency, but we used also 12 features to measure the size and shape. So going from 100 micron almost per eggs to several millimeters in some cases, but also in terms of circularity for the shape. For example, we have very elongated shape in case of bag of eggs, but very round for individual ones. So sorry that everything appears in once. We process principal component analysis in order to search for correlation between those traits. So for those who are not confident with the PCA, the thing is to simplify the information when we have redundant information, because in our case, we have correlated variables. So they are linked, and we want to simplify this data. So we have new axis. So it's called principal component 1 and 2, but behind this, there is our variables. In our case, what we obtain is hue, brightness, size. They are part of the, mainly part of the first component here. Meaning what? That and this part of the graph, we will have small and opaque eggs compared to the other part where we will have big and transparent. And to demonstrate this, this is the corresponding images of all these dots that you have here. And we can clearly see this shift. And also the other important information we have is that the saturation here is clearly behind the second axis, meaning that we have colorful eggs on that coin and pale that. And this is clearly what we see here. After that, so we have one side with small and colorful, the other one with big and transparent eggs. We investigated the phylum dependency of this distribution. So here we find at the center in orange the arthropods, so also part of the copepod are belonging, as I showed you before, with the elongated bags of eggs. The chordates, mainly bony fishes with the big and transparent eggs. The mollusk, small and sometimes colorful. And also part, say, called undefined because we were not able to identify it. So you will say, okay, I said before that I won't show you the taxonomy, so why I'm speaking about this now? It's because there is a pre-identification on the images, so it's only observation. And that's why it's very raw and, let's say, not precise. We also investigated for the fourth objective, the temporal variation. So this is still the same data, but now I show you with a color scale able to show you the temporal, so the time of the acquisition. So in purple, this is the first day of acquisition, and then the other day of the eggs. So meaning that the images I show you here, at least some of them are not just freshly harvested. Here we have a kind of common tendency, and it's a neat tendency for now, that the data and the eggs are moving to a more pale state. And in fact, what we can see with the images here is that on the top of this graph, we have mainly dying individuals. So meaning that there is a kind of opacification during the development. I mean, when they die. So in order to go further in terms of temporal and space resolution, I want to show you a few results now regarding the microscopy acquisitions. And again, we processed PCA. Here, I won't go further in terms of size and shape because each egg is not changing in size and shape by itself during the development. But I want to see the developmental variation. And here what we see is transparency. Transparency because these images were not acquired in color, but in bright field with monochrome camera. And what we observe here is that the transparency is clearly parallel to the second axis, meaning that we will have very transparent eggs on the bottom and opaque one, no, at the top, sorry, and opaque one at the bottom. And if you have seen the previous video, you could have observed that, in fact, the bony fish previously was decreasing in transparency because there is some pigmented cells which are appearing during the development. So this is a first direction, a first trajectory of our data coming from the transparent state to less transparent state. But we observe also another variation in another direction, going from an opaque state to a more transparent one. And this is the case, for example, in some arthropods, when they start very opaque and dark to the light, but just before hatching, they become transparent. So what does it mean, everything? We have, okay, some small and colorful eggs, some big and transparent. So first we have a correlation between the traits, but also it's something already partially described in the literature, that the size and the color are part of a trade-off between the predator avoidance and the UV protection. When we switch to the temporal variation, we observe first opacification of dying individual. So in this case, it's not anymore interesting to understand more about the development itself, but it can be used as an egg quality measurement. And this is already something used, for example, in the industry like aquaculture, for commercial species to assess the healthy state of the eggs. In our case, it could be also used for many different studies. And finally, for the healthy individual, we see also some variation, like I'll show you with the transparency. And here, what is highlighted is, okay, the eggs have some morphological traits, but there are variation during the development, and the aim is also to prepare the larval state. Because here what we have is big eggs at the beginning, so main risk is predation. Okay, so you have to be transparent to survive to the next stage. But the next stage, you are able to swim because this is a larvae. So you are allowed to have pigmentation. And this is the opposite for the arthropod I'll show you here, because they are attached to the mother, like the copepod I've shown you before. So the mother plays a role to avoid predators, whereas it's not the case anymore when they hatch. So they have to change also their trait according to that. So to sum up, we have preliminary results to assess its diversity because we have developed an efficient workflow to record morphological traits, analyze their correlation, and also monitor temporal variation. Now, the taxonomy is currently ongoing because I show you just a pre-identification, but for sure we will be able to go further thanks to the DNA barcoding. So, morphological traits reflect both parental heritage, like size and shape, but also the physiology with the color and transparency, for example. And it's a powerful tool for integrative studies, both in plankton ecology and developmental biology. And the rest of this project will be to analyze embryo traits that we have also extracted in the images and process also the images again, this time searching for developmental traits, like number of division, at which time the division, in which symmetry or axis. And finally, the quality, the egg quality could be go further, more in terms of ecology. And automation of this system will be done also to provide some tests in machine learning and be able to implement this data in databases. I thank my supervisors, Michael Schubert and Fabien Lombard, but also Jean-Yves Carvel and Pierre Cohen, because without them, I couldn't have any sampling and sunset every morning. Thank you for your attention.
Thank you, Maeva. Thank you a lot. And congratulations for your presentation, despite the surprise with technical aspects. So now we're entering in the phase of questions. You have here 10 jury members. If you want to reactivate the thing. So I introduced you to the different jury members. Aldine Amiel. So from IRCAN, we have Aldine Amiel, Paola Furla, and Eric Rottinger. IRCAN is the laboratory from Université Côte d'Azur. Aldine Amiel is research engineer, specializing in developmental biology, regeneration, cellular and molecular biology. Paola Furla, professor on coral reef ecology, marine biodiversity, cell culture, molecular cell biology, and others. Eric Rottinger, I took that on your Google Scholar thing. I'm sorry. Eric Rottinger is researcher still at IRCAN on biology, regeneration, developmental biology, cnidarians, and aging. I think you know that. Yasmine Botin is professor at Thesis, another laboratory at Université Côte d'Azur, specializing on conservation, environmental toxicology, international organizations. Still from UCA, but with INRAE, Elena Bovio. Here is the postdoc at INRAE, specializing on microbiology, marine natural products, fungi, and bioremediation. Online also we have as a neighbor Stefano Tiozzo from Sorbonne, IMEV from Villefranche, researcher specializing in developmental biology as well, regeneration and pluripotency. Hello Stefano. Robin Fayetaz and Anne Pajot are both from Ifremer. Robin from Lorient. Robin is researcher on fish behavior, selectivity, biophysical modeling, and imagery. Anne Pajot from Brest is a PhD student on microalgae, process engineering, and microbiology. Two more. Letizia Zullo online from Italy, from the Italian Institute of Technology. Hello, Letizia. You are a researcher on neurobiology, robotics, physiobiology, and Octopus. And finally, Jean-Vincent Vieux-Ingrassa will come with an external view. Jean-Vincent is a teacher, a biology teacher in high school. And he will be here for the 3 sessions as external point of view. So I will propose each of the different jury members to ask your favorite question. One, so you can try to prepare a few in case the others took yours. So I will start with Aldine, maybe. Aldine?
Yeah. So thank you, Maeva. Really nice talk. So my question is, so how do you plan to link the barcoding work with all the feature analysis you already performed?
Can I just keep them out? No?
Pas la question, garde, je suis désolé.
Okay.
Aussi avoir ça.
Okay. So the plan, once we will have all the sequences, and in fact, it's not true to say all the sequences because I have not the sequence of all the images I show you. So it's partial. The aim will be to represent, as I show you with the phylum case, to represent this data in color. And also, because we won't reach the same taxonomy level, we won't reach all the species, for example, maybe we could process step by step the analysis with, for example, the class and then the family. And if we start to see some pattern, maybe we could also process some clustering analysis. I don't know if you wanted me to go further.
No, it's okay. Thank you.
Okay. Can we continue with Paola? We'll take some order.
Okay, I have some questions about your PCA. I would like to know how you can define the robustness of your groups, the colors groups, and also the temporal groups, and linked to that, how you have decreased the collinearity or the correlation between your variables.
First, the group that I've shown are arbitrary group. I mean, I show you the color really corresponding to the pre-identification, but the circle that I show you is not part of a cluster analysis at all, because for now it was not the aim. The aim was just to see if we had a distribution which looks random, or if we could recognize maybe a separation according to that. But because we were expecting more detailed analysis, we didn't go further this. And how we decrease the number of variables. First, okay, I didn't show all the eigenvector here, for sure, and even in the report, I guess. It's more in terms of size of this vector. And also, what I show you here when I say, for example, size, in fact, behind this, there is also many variables related to the size, like the area, the perimeter, and things like that. So it's a group of variables going in one direction, and I took the mean direction of this vector to show you these results.
Okay, so you took the mean of the different vectors, or you choose one vectors between different ones?
For the size, I've shown the mean in terms of direction, but this is really the size of the eigenvector itself. I mean,
okay,
size is, how can I say? It's not defined by size. It's area, again,
okay, shape.
No,
it's another fact.
Yeah, in fact, they are linked, clearly, but the shape will be assessed with other things like circularity, aspect ratio, width. So some of these variables are linked to the size, for sure, but they can represent also the shape.
I understand the second part of the question. I don't understand really why you say that you don't want to do groups, because you show us the groups, and it's one of your take-home message to have these different groups that you can distinguish.
Yeah.
So I don't understand why you say I don't want to do it.
To show this group with the clustering, maybe?
Yeah, the clustering.
The aim, in fact, is because we didn't want it to process it without the clear genomic data. Because if I want to be very robust, it's observation and it's biased because this is according to already defined morphological traits that I have identified them.
Yeah, but the aim is really to see, may I have some very similar eggs belong to different groups, phylogenetical groups, like chemical groups.
Yeah.
So it will be interesting to see after if you have this correlation between the clustering and your taxonomy.
Yeah, clearly. But in fact, we didn't have the time to go in this direction, and we thought, okay, maybe the best way would be to have first the DNA information to be more strong on this.
Thank you.
Paola. Eric, do you want to continue?
Sure, Maeva, thanks for the presentation. Nice project. Very quick question, and perhaps you mentioned, but I may have missed it. But measuring colors is always very tricky. So do you have a calibration step so you can compare the colors between the samples? I didn't get that.
Yeah, I didn't present it now. I explain it in the material and method in my report. Okay. So the first thing is the color. Here you have seen some dark field images. So it's already not the perfect way to assess color because, as you may know, it's a diffracted light that we are clearly seeing here. So yeah, I had the head of the authors. And the thing is, in fact, we didn't expect to acquire all of this data. So in order to calibrate them, I have done a kind of black background correction. You know, like a black white balance would have been done. I've done the process on the opposite way to correct the black because we had some variation in the acquisition of the images. And also to be more robust, I avoid to count in the region of interest the border because this is where we have a high diffraction of light. And so the color that we could assess could not have been the real one. So I reduce a bit the area of study.
Okay, thanks.
Thank you, Eric. Let's continue with Yasmin.
Yes, thank you for the very nice presentation, despite this unexpected type of videos. So I noticed one sentence you said in your introduction that there was a link between the shape and the environmental stressors. So with all those phytoplankton that you have analyzed, what would you expect or project as changes in the overall trait of the plankton with environmental stressors, thinking of increase of temperature or any climate change? Do you have any thought about that? Or?
Particularly linked to the shape or all the traits?
Any of the traits, yes.
So it starts to be documented. The first one important study was done not in terms of environmental stressor, but just environment. And they have seen some different patterns in terms of eggs depending on the different oceans. And then they explained, but they didn't investigate it more. And it could be because of the light variation, because they have focused the study on pigmentation, which provides some more opacity and color. So it's an idea, because with the climate change, we could expect, not climate change itself, but it's also predicted to have more UV exposition due to the changes in the ozone. So it's a way, but also in terms of acidification, temperature, it can affect the size because what is already described is, at least for the adults also. From a project, I focused on the eggs, but we could also extend this to the larval stage when appears also the defense structure outside. And this could be made with some minerals like carbonate or silicon, which for sure will change with the global change. So this is a kind of way. And it's something that ecologists are very also interested in to see even at a smaller time scale between the season. And it's already something that appears, at least for the adults, there is some variation in terms of pigmentation according to the season to adjust also with the metabolism and energy available.
Elena?
Okay, thank you for the presentation, and I was one of the viewers, but it was a really nice job and very interesting. And I wanted to ask you about the method of the barcoding, sorry. So I've seen in the supplementary material that one of your sequences matched with uncultured eukaryotes. So how would you progress when I find something like this?
So when it's not supposed to be
yeah,
the egg, yeah. So there is the easy case where because I plan to sequence two genes, one in 18X and one in cytochrome oxidase 1. So if I have only this kind of results for one sequence and the other one give me something more trustable, okay, we could say that maybe we could use only this data. The other thing is that maybe we could use other sequences, but the problem is to find corresponding databases then to identify it. And it's something limited in all the barcoding analysis currently now. And in our case, it's very tricky because we have also noticed that many parasites are present in the eggs and disturb really the development. And even with all the care that we could do to the sample, we are not sure that we won't amplify the DNA of the parasite itself, because it sequences in common also with them.
So you are facing something new. It's impossible to go further. I mean, to describe maybe a new species or something like that.
No, no, I didn't say it's impossible. It could be interesting. For sure, we won't use this currently in this analysis. But then to go further, it could be interesting. Then in the future, maybe to harvest, to maintain more also this species, to use also other sequence to find if maybe someone sequence another part. So search also other sequences. And if not possible, it means that all the work needs to be done. And in this case, we need to have the chance to culture it until the adult stage.
Thank you.
Thank you, Elena. Stefano, maybe?
Yes, thank you, Maeva, for the nice presentation. And it seems that you collected a lot of information, so it's a good work. Well, I have several questions, but I guess I will ask one or two maybe. One says, I'm not sure if I understood, when you talk about your objective 4 and the temporal variations, are you talking more in the developmental? So you are looking at the eggs at different time points, or did you collect at different time points?
No, with the data that I have currently, it's at the developmental scale. Then the aim will be to develop an automated system, like for example, we use already with SoScan or FlowCam, in order to have also temporal acquisition over years, and in this case, to observe also seasonal variation, yearly and meteorological variations.
Yeah, that brings me to another question. So already the parameters that you are looking are well, a lot. They are representative. You choose. You have your reasoning for choosing. But if you sample if you sample at different time of the day and according to the meteorological, so I assume you will have different results. So did you think of a way to kind of overcome or flatten this huge delta that you may obtain in your data set?
In terms of daily, daily
variations. In terms of variations in the daily or circadians, or why did you pick? I assume that you pick the collection time when the boat was going out in the morning. But
it's not. The boat is going out also in the afternoon,
yep.
And despite this, we didn't use this time as this slot.
Okay.
In fact, we preferred the morning because most of the currently described species are releasing eggs during the night to avoid predation and let the eggs disperse before that predator are going up again. So we expected to have more eggs at early morning. Then during the day that they could be even more dispersed and eaten. However, it's something also that I discussed, because I would suggest to try to have several sampling within the days, if we put on the side the technical and feasibility, but it will be very interesting to do it at least on a short period to see the differences, because there is also some species releasing the eggs during the day because they have other strategies, and also because we can have a huge variation due to currents, and it's not something that we have assessed for this study.
Just one quick technical question. So for the collecting, for the net that you choose, you wanted to look at which range of eggs? Because you have to make a decision there. You have to make the decision there too, right?
Yes, I have tried all. So from the phytoplankton, WP2 and regent. The problem is then the efficiency of the collection. For sure with phytoplankton, because you can gather individuals from 20 micron, you could assess that, okay, I have all of them. But then how to sort them? Because you have also all the phytoplankton and you lose so much time and the eggs are so degraded at the end. So it's not good. And the regent is more protective for the individual. When you collect, the problem is you have only fish egg, because it's from 680 micron. So for me, it was the best compromise to use the WP2 and in vertical way to have a kind of trade-off between, okay, I can collect almost all metazoan eggs without so tricky sorting.
Thank you.
Thank you, Stefano. Let's move to Robin. Hello, Robin.
Hey, I'm here. So I'm just going to ask a quick and more general question. Everything, I was a reviewer also, and I really enjoyed it. It was super interesting. But everything you talk about is about morphological traits, because you didn't do any taxonomy. I mean, you tried to do, but you didn't get the result. I completely get it. But given your results and like the different groups and like all the PCA and the distinction you have, the morphological differences you have between the groups, what do you expect from the importance of the taxonomy versus the morphology in the context of climate change? Like, is the actual morphological traits going to be more important than the taxonomical trait in understanding the effect of climate change? And what do you think the taxonomy will really bring something? Do you expect more variability between the groups, between species, between morphological groups? What do you think of that?
I think it's complementary, because if we only use morphology, we could avoid taxonomy if we focus, for example, on functional communities. And maybe we could already assess some data according to changing in environment. But we could not be sure 100% it is the same species which react to its environment and change something. And also because the aim would be to spread this method, now it's obvious for many plankton scientists that we can use Zooscan, for example, or any UVP. We could assess also same species in different environments. And if we don't have the taxonomy, we could forecast what could happen for the same species in different environmental conditions. So I think both are clearly complementary, and it depends on then the question, but the aim would be to predict the climate change and how the species will react. So we clearly need the taxonomy now to find if the species are reacting the same in different environments. To my point of view.
Thank you.
Thank you, Robin. Thank you. Let's continue with Anne Pajot. Hello, Anne. Hello, Anne.
Yes. So I have several questions, but I will only ask one. Do you already know if the 48 features, if I well understood your abstract, allow the precise assignment of species to specific taxons? Do you already know that? But I know it's in progress, so.
Yeah, it would have been the golden objective, to then acquire enough data, maybe process machine learning, and then have, okay, this is a morphology a description, and okay, now we have enough data to say this is this taxon. But it's not the case. I mean, the number of potential species that we don't know in the plankton is huge. So it won't be enough. But because we don't know yet what are these data, it's possible that some groups could be identified by this way. So maybe not at the species level, but maybe a group or a family of individual could be. And this is also what we are searching.
Okay, thank you. And thank you for your great presentation. I really loved it.
Thank you.
Thank you. And Laetitia?
Yes, hi. Thank you for the presentation. It was really nice and very interesting work. I have a question about what you showed in the very beginning about the possibility of a marker of parental investment. So I would like you to explain what exactly it means, a marker of parental investment.
Yeah. So what we call parental investment is how the energy cost is involved for the production of the eggs and of the future generation. And it can be assessed by trait like I just mentioned, number and size of eggs, but it's also something in terms of reserve that will be given to the eggs to survive. And it starts to be documented because we have already observed that according to the food that, for example, the mother can find in the environment, we don't find the same content in the reserve of the eggs. So sometimes some stressed mother couldn't provide some pigment to their eggs, and so their chance of survival are decreased. And we know that it's also a huge cost because there is also some very few species when we observe a kind of choice of phenotype for the eggs. And for example, there is I don't remember exactly the name, sorry, I have it on the slide, that a benthic species, the mother has the choice to release the eggs or keep it in. And the eggs in are not pigmented, whereas the eggs which are released are pigmented. And the same individual is able to do the both according to the environmental condition too.
Okay, that's very interesting. Thank you.
Thank you, Laetitia. We will conclude so with Jean-Vincent.
I have a question about the choose of morphological traits to create functional traits.
Okay.
What are the morphological traits to that you can choose to create functional groups?
Alors, at the egg state, it's more in terms of, I would say, there's several possibilities. It could be, as I've mentioned, the small and big, because it's something used already for the adults. We know that, for example, with copepod, we don't, the function does not rely on the species itself, but more on the type and size of the copepod. So maybe it's the same for the eggs, but we don't know now, so we should investigate this. And also, because the eggs provide a huge source of feeding for many species. So the thing that we don't know is that either a particular type of eggs, which could be preferred maybe to be eaten. And then I think we could find also things later, more in terms of larvae, which are able to spread and swim or not, and maybe also in terms of their migration.
Thank you, Maeva. That's it.
Okay.
Congratulations for your presentation. Frankly, the jury appreciates it. And so see you soon. We will continue in a few seconds. Bye, Maeva. We will continue in a few seconds with Ludovic, who I'm sure is ready. Okay.
Pourquoi je ne le vois pas?
Gesson, tu vois pas Ludovic, c'est ça?
Enfin, je le vois là, mais il n'apparaît pas sur la plateforme. Et j'avais vu que tu ne l'avais pas mis sur 2 sessions. Tu sais, je t'ai envoyé un message.
Non, un instant, ça va pas prendre long. Oui, c'est par personne. Normalement, c'est bon. Je vais rafraîchir là. Okay, Ludovic, did you try the remote? Does work? Okay, good. Don't worry, I will remove you from the screen behind you. Okay, it's all fine now. And you can start when you want if you have time with you. Okay, good luck.
Is it possible to have the PowerPoint?
Yes, in a second. Thank you for reminding me that. Just 1 second, so before you start, can you move a bit on your right or on your left? Yeah, thank you. It should work.
Okay. So everybody can hear me correctly, everything is fine. Okay.
Let's go.
Okay. Hello everyone, today I'm going to present to you my master thesis subject that I realized in the laboratory of Villefranche under the supervision of Jennifer Cross. So my host team has been mainly working on the animal nervous system evolution topic, and to address this particular topic, they've been looking into the adult nervous system of an echinoderms, the sea urchin. So my master thesis within this research topic aimed to answer the question, how does the adult nervous system of Parasamphotis lividus form during rudiment development? And here I'm going to give you a quick introduction about what is the nervous system. So the nervous system and function is to give control to the organism in order to be able to collect, process, and respond to sensory stimulus. The nervous system is also composed of cells, named the neurons, that can be really easily identified by the cellular components, such as the cell body, from which an axon will extend. The axons are made of a protein named microtubulin, which will ensure the maintenance of the structure, as well as carrying out the neurotransmitter. At the end of the axon, you find an area named the synapse, where the neurotransmitter will be released and therefore transmit the neuronal information to adjacent neurons. So here, it's all you have to know for this presentation. And now we are going to look at what are the echinoderms. So the echinoderms is a phylum of organisms composed mainly of only of marine organisms and divided in 5 groups, including the echinoidea, in which I had a big interest for my master thesis. They are also defined by synapomorphisms, including the pentaradial symmetry that you can really easily see for the sea stars, but is also present for the sea urchin, and also the locomotion mode, thanks to the water vascular system, that you have a little example here. But if you want to study echinoderms, we also have to understand the life cycle and development. So here, I took the example of my model species. And my model species display an indirect life cycle, as most echinoderms do. So what does it mean for an organism to have an indirect life cycle? It means that during the life of the organism, there will be 2 stages. The first one, a pelagic stages, with a larvae that goes from the embryogenesis to the end of the larval period. And after, we have a benthic stages with the juvenile and the adult. So what also means for the organism is that during its lifespan, it will possess 2 distinct nervous systems. The first one being a larval nervous system. and the second one being an adult nervous system. So historically, the larval nervous system has been really well studied and characterized both anatomically, molecularly. So characterization has been established. On the other hand, the adult nervous system has been less studied, and currently there is no complete characterization. Therefore, if we want to bridge this gap of knowledge about the adult nervous system, we have to understand the development of the adult. So here, I retook the life cycle of my model species, and here, this is where happened the development of the adult. So if I sum up it really quickly, first, you have the formation of the larvae, and during the larvae development, in the left side of the larvae, a new structure will develop called the rudiment, and the rudiment will be the precursor of the juvenile. Once the rudiment reaches maturity, the larvae can undergo metamorphosis. During the metamorphosis, the rudiment will evaginate, engulf the larvae, and give rise to the juvenile. So if we want to study the adult nervous system development, we have to take a look at this particular time frame. And also, we have to use a model species. And for this case, we took Paracentrotus lividus. So why have we chosen Paracentrotus lividus as our model species? First, it's a local Mediterranean species. So the organisms are really easily available from Villefranche-sur-Mer to collect. Also, the lab has been successfully developing a husbandry protocol. So we are able to generate a lot of organisms for the experiment. And after, the lab has also successfully developed working nervous system staining protocol for the juvenile. And thanks to those working nervous system protocol, they've been able to do the first characterization of the juvenile neuroanatomy. And here is their results in terms of neuroanatomy. And they've been able to highlight 4 major nervous structures. The first one being the circummoral nerve ring here, 5 radial nerve cords, the podial nervous system here, and the spine nervous system. However, this first neuroanatomy is not yet complete because they are still missing the adult nervous system development before the metamorphosis. And therefore, come my biological question. How does the adult nervous system of Paracentrotus lividus form during the rudiment development? And to address this question, I have 2 different goals. First one being to do an anatomical characterization of Paracentrotus lividus rudiment development. And the second one, doing a molecular description of the developing adult nervous system within the rudiment. And here, I'm going to expose you the experimental approach we took to address this question. So first, we launch a different set of culture of larvae. Once the larvae reach an age between 7 and 25 days post-fertilization, we're able to use the larvae for the experiment. And for all the experiment, we focus on the rudiment only. The first experiment we performed was the rudiment anatomical development characterization thanks to image acquisition. After, what we did was the immunohistochemistry assays in order to be able to select several molecular markers of the nervous system. In our case, we selected only the one that were staining the nervous system within the rudiment specifically. So we had the acetyltubulin, which will mark the neuron axons, and we have a neurotransmitter, the histamine, which will stain specifically the histaminergic neurons. And thanks to those 2 molecular markers, we're able to do the adult nervous system characterization. So here is the first set of results that I'm going to present to you, which is the full rudiment anatomical development. And I'm not going into detail with everything that you saw on the screen, but what you have to remember is that thanks to the image acquisition of the rudiment development of Paracentrotus lividus, we're able to distinguish 13 distinct stages using different anatomical criteria to distinguish one stage over the other. And under this new timeline of the rudiment development, we're able to select 5 stages to investigate the adult nervous system development. So speaking of adult nervous system development, here is the first result that we obtained with the acetylated tubulin. Just quick note, the acetylated tubulin has been previously used with the juvenile neuroanatomy characterization. And after, we have the results with the histamine that has never been performed before on the adult nervous system of Paracentotus lividus. So what is the take-home message for this result? This is that we were able to observe the establishment of the four nervous structure that has been previously described in the juvenile neuroanatomy. So the circumoral nerve ring, radial nerve cord, podial nervous system, and the spinal nervous system. And for this presentation, I'm only going to focus on the podial nervous system. So first, we are going to look at the podia anatomical development, and after, I'm going to show you the podia histaminergic neurons development, because I think it's more interesting because it's never been done before for this species. So here, if we take a look at the first stage of development of the podia, the five-fold ectoderm stage, you have a little cartoon here to help you understand the orientation of the picture. And here it's a lateral view. So here, what are we looking at the five-fold ectoderm stage? We have a folding movement here. And if we take a look at the next stage, the primary podia, we can see that the folding movement is extended and lead to the formation of the podia. Now, if we take a look at an axial view for the podia touching stage, we can see that the podia are not touching at the epidermis at their tips. Finally, for the twisting podia stage, we can see that the podia start to twitch due to the growth and the lack of space in the rudiment. For the next stage, the papilla stage with the axial view, we noticed the formation of a new structure at the epidermis at their tips called the papilla. And finally, at the combatant stage, the podia were fully developed. And what does it look like? The podia fully developed. You have here a closer view. So we have a stem fully developed and the presence of a papilla. So within this podia anatomical development, we were going to investigate the histaminergic nervous system formation. So here we have the first stage for podia touching, which was quite similar with primary podia, and we were not able to identify histaminergic neurons. However, the twisting podia stage, so the next one, we were able to identify some histaminergic neurons within the podia epidermis. Here, if we take a look at the papilla stage, first, we're able to identify the papilla and the stem within the picture, because they were formed, as well as some histaminergic nervous system structure, two circumferential condensation, one at the papilla and one at the papilla basis, and histaminergic neurons within the epidermis of the stem. And if we take a look at the last stage, combatant stage, you can see that there is a lot of new structure that have developed, such as, so we are able also to identify the papilla and the stem. The first histaminergic nervous structure I'm going to highlight are some histaminergic neurons that connect the podia epidermis to this large structure that we call the large histaminergic nervous condensation. And after, these histaminergic neurons within the stem are forming a kind of net. So what's really interesting about those results is that when we look at the literature, we're able to identify that the podia and histamine has been independently reported to have reported to be involved into some environmental sensing. Therefore, when the environmental sensing is really key for the metamorphosis, because the metamorphosis can only be triggered when, one, the rudiment has reached the competent stage. Therefore, it means that the podia will be fully anatomically developed, and therefore it will have the stem and the papilla, and also it will show this histaminergic nervous system. Once the rudiment reaches the competent stage, the larvae will swim down to the seafloor, and one podia will touch the substrate. And the main theory about that is that the larvae is able to sense the settlement cue. So the settlement cue are the environmental factor that will trigger the metamorphosis with the podia touching the substrate. Therefore, the fact that podia in histamine has been independently reported to be involved into the homotal sensing, and we found at the competence stage this condensed histaminergic nervous system could corroborate the podia sensory function. So, for sum up a little bit everything I presented to you, we're able to identify the podia anatomical development with different stages. Each of the stages has their own anatomical criteria to distinguish from one another. And during this time frame, we're also able to highlight the histaminergic nervous system formation with the apparition of the different structure within the different stages. So in terms of conclusion and perspective, so my work has constituted the first characterization of the rudiment development and adult nervous system formation of Paracentotus lividus. Here I only show you the podia part, but the definitive spine, for instance, has been investigated and other structure. Also, it helps us to establish a new developmental timeline of Paracentotus lividus rudiment development. Also, for the histamine, it was never performed before on the adult nervous system of Paracentotus lividus. Therefore, it shows us that it can be used for studying nervous system formation. And thanks to the literature review, it could be used for assessing the podia sensory function. Finally, my work helped to gain insight about the echinoderm adult nervous system. In terms of perspective, we could deepen the characterization of the nervous system development with gene expression and functional analysis. And finally, it will be really interesting to compare the adult nervous system development of Paracentotus lividus with other echinoderms. And I would like to thank you for your attention.
So we'll come back on the other way for the questions this time, because it's not easy to start always with the questions or to end with them. So maybe we can start with you, Jean-François.
So I have a question about your guidance animal nervous system evolution. So maybe I have a question about if it's possible to use the steer genes as a model to explore the evolution of the nervous system through the evolutionary point of view.
Yeah, definitely. In fact, so I explained the first slide that my host team was really working on the animal nervous system evolution topic. And in fact, there is, for the animal nervous system evolution, there is really one big question, which is when those decentralization events of the nervous system appears. So in the animal kingdom, there is really two main categories of nervous system. The diffuse nervous system that you can find, for instance, with the cnidarian, and also the centralized nervous system. That's, for instance, the vertebrate process with the anterior condensation, the brain, one or several longitudinal cords. So for us, it's the spine and peripheral nerves. And the big question was to understand when those decentralization event appears. So does it appear once in evolution? So like the ancestor process is centralized, or several times? And the big way to answer this question is to look at alternative nervous system, like the one from the sea urchin, that doesn't look like the centralized one. But if we go more deeply into the analysis, maybe they are quite similar. Therefore, it could answer the question, like the question about decentralization events. So it's quite complicated, but it's really looking into the alternative nervous system and look how it's referred to the centralized one.
So it's possible a good model?
It could be, yes.
Thank you, Laurent. Laetitia? Laetitia? So, yeah.
Hi. Okay, thank you for your presentation. I also was a reviewer of your thesis, which I enjoyed a lot. I have few questions, but I'll ask only one. So what you want to do is to study the role of histamine during development. And I'm wondering whether you have, since histamine in some other invertebrates has found to have an inhibitory action, so to be used as inhibitory neurotransmitters. I wondered if you have an idea on what could it be, the role of histamine, the functional role of histamine, and also if you have considered the possibility that some of the staining you might obtain it's due not to histamine used as a neurotransmitters, but histamine that is produced by other cell types.
Okay. Yeah, I saw what you mean. So for the sea urchin, in particular, so the Echinoderm, the effect of histamine in development can quite vary. Like there is some species where the histamine will be necessary to achieve the competent stage. So it will have the rudiment to acquire this particular stage, but it will also inhibit the metamorphosis without the settlement cue. And for other species, it was quite the opposite because the histamine was the settlement cue. So it was really like inducing the metamorphosis. In our case, we didn't do like pharmacological tests to see whether or not the histamine could have an effect on the development, but it could be interesting indeed.
Okay, and do you know if it can also be produced by other cell type in your model, of course?
In my model, I don't know. It could be if my model possesses like more immune system,
yeah,
like an answer, but yeah, we didn't investigate this part.
Okay, okay, thank you.
Thank you, Laetitia. Anne?
Yes, thank you for your presentation. I think it completes very well your manuscript. I reviewed it and it was pretty good. And I had a question about it. Why do you induce the metamorphosis of your echins? Is it to accelerate the process? And if so, how are you sure this acceleration doesn't have consequences on the nervous system development after?
So in our case, we are just looking at the system nervous development during, like, until they reach the competent stage. So we're not like interesting about the after effect, let's say. And we induced the metamorphosis because it's more easy for us to like obtain juvenile, because they can like, in our culture setup, like for the metamorphosis to happen, they need like the substrate, they need the algae. So it was kind of hard to reproduce those conditions in our like culture setup. So what we used was a substance called the DBM, which is in fact chemicals that are found in some algae that are on the seafloor and that have been linked to the metamorphosis of the sea urchin.
Okay, thank you.
Thank you, Anne. Robin?
Nice presentation. So this is very far from my project, so from my topic, so it's maybe again question, but I guess the neural system for that is linked to the swimming is only during the larval stage, but you studied the development of the podia system, how the two interact, if they do, and is one, I don't know, is one developing, because I guess the larvae will be swimming before the beginning of the development of the podia system. So how do the two interact, and do you think there's a link between, I don't know, the metamorphosis and the disappearance of the swimming? I mean, the neural system associated with the swimming.
So if I understood well your question, is there a link between the larval nervous system and the adult nervous system in the metamorphosis? Something like that?
Linked to the swimming behavior, because the larvae would be swimming, and because the larvae to settle, it either will stop swimming or will start swimming down. So I guess that there has to be an interaction between the sensing of the environment and the choice of the decision of the changing behavior of the larvae to settle.
So first, there is no like clear evidence for communication, but the larvae cannot like undergo metamorphosis without the rudiment to be totally developed. So therefore, it could be a connection between the two in order for the larvae to be able to know when the rudiment is like at maturity. And also, the one hypothesis is also that the larvae is able to sense the environmental cue and will swim down to the substrate, and the podia touching the substrate will confirm maybe this environmental cue to trigger the metamorphosis. Like larvae, when they reach competency, will just swim down, and if there is no cue, they will just go like swim back and in the water column.
So the competency is reached once the neural system, the sensing neural system is mature.
Yeah. And I also have been looking at the formation of the histaminergic nervous system within the larvae, and the development of the histaminergic nervous system in the larvae also appear around the competency. So both have those like histaminergic nervous system that help them to sense the environment.
Thank you.
Thank you, Robin, pardon, Stefano.
Well, thank you very much for the nice presentation, very clear. So my first question that you partially gave an answer to that, but I'm still not convinced. My first question was why histamine among the other neurotransmitter that you could choose, because one of the problem that also Letizia raised is that histamine has an important role on evolutionary speaking in the immune system, and it has been seen in several marine invertebrates. So how do you know that what you're looking at are neurons? Did you do a double, for example, double labeling with tubulin, or?
Yeah. So first we selected the histamine because it was one of the only antibody and molecular marker that were staining really specifically the nervous system within the rudiment. Like we tested a lot of other substances, but they were never like staining specifically this nervous system. Therefore, we could not study the development.
So you didn't stuff like serotonin didn't work, GABA or cholinergic.
Like we tried ephemeramide, glutamate, we tried noradrenaline also.
The usual suspects.
Yeah, and it was like we were happy to have like one that was working, but also we selected this particular one because of its like importance in the metamorphosis. So therefore, it's linked to the life cycle and the adult development. So we were thinking it was quite interesting to see if histamine could play other role that like inducing rudiment development and inhibiting the metamorphosis.
Did you try a double stain with the tubulin?
Yeah, when we tried, we did with the tubulin to see if it was working, and we did also the control, where we're only using the first antibody and the secondary antibody to really be sure that we were specifically staining the neurons.
And just one more broader and evolutionary question, because since evolution is in the title of your presentation, and so how much is it known about the diversity within echinoderms in terms of nervous system? In the post-metamorphic, but also in the planular larvae, is it pretty robust, or?
It's quite at the start, let's say, but the first results show that, for instance, the sea stars, the ophioid, and the sea urchin have the same anatomical features in terms of the adult nervous system. So the circumferential nerve ring, the 5 radial nerve cores, and the peripheral nerves. So in terms of anatomy, it's quite similar. And the next stage will be really to look into more detail into the development and all the genetic analysis to see if it's quite similar as well. But it's really at the start.
Thank you.
Thank you, Stefano. Elena?
Okay, thank you for your presentation. I'm a little far from the topic, so, but I have a question. You work in the lab, so you say that it's a nice model. And do you think that what you observe can be applied to what happened in nature, and also if you know, if climate change might change in some way the development of the nervous system?
This is one point. Like I was doing really fundamental research, because like if we want to know if like climate change has effects on the development of the nervous system, we have to understand what is the nervous system development. So here, yeah, we could, from our knowledge, try to see if there is like impact of the climate change, like pH increase or temperature increase on the development of the nervous system. So it's really the start of acquiring the knowledge to be able to, after doing like environmental testing and different experiments, to test the different scenarios.
Thank you.
And Yasmin?
So thank you for the presentation. I don't know if I have missed it, but I would like to have more information on the number of replicates you have used and how, and have you seen any variability within individuals when you are staining and you find always those 10 stages?
Okay, so I had like two sets of results. I have the first one with the anatomical development. So what we did is that we launched different sets of culture, and like every day we're like taking picture of different larvae at different life stage from different culture. So I don't have exactly the number, but we were like trying to not use one individual to do all the picture, for instance. So we had a little bit of variability, but we looked at all the picture and tried like to find the correct stage for each of them. And also we use previous like literature to help us to understand the different stage.
So those stages are also found in other organisms.
Yeah. What we did is like we tried to like give more precise stage. Okay. But it's really important to understand it's more like qualitative analysis and not quantitative.
Yes, but it's important to know if those stages are systematically seen or if it's just.
Yeah, it comes from several cultures from like a lot of different larvae.
Okay, thank you.
Thank you. Eric?
Hey Ludo, thanks a lot for your presentation. It's good to listen to a CHN talk. It has been a while. I have actually a question that you sort of started to respond to in answer to Robin's questions, but it was not the swimming behavior. My question is, to what extent the nervous system of the larva, particularly of the rudiment, is integrated into the nervous system of the adult? Or is it an apoptotic event and then everything is done de novo?
Okay, so during the metamorphosis, the larvae will be have the apoptotic stage, and the only nervous system that will remain is the one in the rudiment. So it's really created de novo.
But so the rudiment that grows and becomes a sea urchin, at the beginning it's the solemnic pouch, sorry, but it's the beginning it's the solemnic pouch and it is already nerved, right, or not? Or is it devoid of a nervous system at the larva stage?
Sorry, I didn't understood correctly.
The solemnic pouch that is at the origin of the rudiment, is there already a nervous system that is part of it and that it grows afterwards or not? So I misspelled actually what I meant.
Okay. So when the rudiment is created, it's like the fusion of the vesicle and one scleromic pouch, and there is no trace of nervous system within this pouch that is in the larvae. So it's really created de novo.
Okay. And then I have a quick other question. You said at the end that it would be interesting to compare to other echinoderms, and you mentioned over and over again. If you have to choose one or two other echinoderm species, which one would you choose and why?
Okay, I will choose the crinoid because of their basal position in the Echinodermata tree, and it will help us doing the comparison between the crinoid and the sea urchin to like rebuild the nervous system of the last common ancestor.
Okay, thanks a lot.
Thank you, Eric.
Hello, Ludo. I think that most of the questions have been asked, so I try to focus more on the podia and the link with the nervous systems. And I would like to know if there is some podia on the echinoderms, on the different size. I think more about the sisters, that I know that the very far podia are very sensory, they are quite long, they are less using for the locomotion, that more with the sensory function. So my question is, did you see, or do you think that we can see some differences in the organization of the nervous system in the different part of the ambulatory systems, or everything is the same?
So from what I read in the scientific literature, it was more all the same for the podia in the sea urchin. Okay. Like they present like photoreceptor, mechanoreceptor, chemoreceptor, and for the photoreceptor, it's like on every podia. Every podia, yeah. Okay. Doesn't mind they're close to the mouth or very far away, it's the same. There is no like a differentiation like in the system. Okay.
Thank you, Paola. We will conclude with Aldine.
Hi, Ludo. Thank you so much for your presentation. I have a question related to also evolution and so on. But you are working with complex, very complex systems. So there is sea urchin that have direct development. So why you don't choose such system for your biological question?
So yeah, there is like direct developing sea urchin, but in our case, in our lab, when like there is no model that have been developed with this developmental life cycle, and also my model species, so Paracentosis lividus, has been well studied before, has been a model species like for the development of the larvae for the different stages, such as the blastula, gastrula, has been studied very deeply in the larvae. So like we could use all the knowledge about this biological model now compared to another species which display maybe a more like relevant developmental mode, but we don't have all the knowledge, so we really have to start from zero.
Okay, and regarding the presence of the larva in sea urchin family, do you know if the ancestor of the sea urchin may have the larvae in their stage, or if it was a direct developer? Do we have some insight about that?
Yeah, not with like my master thesis result, but from the literature, the main theory is that the ancestor of the echinoderm should have larvae.
Okay.
And it will lose in evolution.
Okay, thanks a lot. Thank you.
Thank you for the jury members, and thank you, congratulations, Ludovic. Good luck for the followings.
Thank you.
Soon. I'm afraid we have to continue before break. We have 6 presentations today. I propose you to do a break after the next one. Okay. Okay. Sinon, on fait plein de pauses. We will continue with Hugo Allazio, who is going to talk about deciphering the sensorimotor control organization, the Octopus arm. For Hugo, Laetitia was part of his supervisor, so of course, Laetitia, you will not be part of this jury. Just asking for the jury online if you can see the slides of Hugo.
Yes, we can.
Thank you. So go. It's your time. 15 minutes.
Thank you.
Do you have time for this? Okay. Can help.
Okay, thank you. So first, before beginning, I want to apologize because I have videos on my presentation, and this is the PDF of the presentation, so there will be no video displayed here, but there are illustration videos, so there will be no lack in the information I will present you. So I'm here to present you my thesis, which was deciphering the motor control on the octopus arm.
Yeah, try again.
Okay. So this presentation will not follow a common outline. I will present you the host institution, the context, and the goal of the thesis. Then we'll go through the results of the three aims of this thesis with a presentation of the result and an explanation of the results. Then a general discussion and future direction of the project. So I did my internship remotely with the Italian Institute of Technology, with the laboratory based in Genova, which is the Center for Synaptic Neuroscience and Technology. I was supervised by Letizia Zullo and Alessio Di Clemente. So let's start with the study subject. So we studied the octopus. The species was Octopus vulgaris, which is a cephalopod from the phylum of the mollusk. It is well known for its 8 arms, its really soft body and high flexibility. And despite having the most centralized nervous system of all the mollusks, it has 2/3 of its 500 million neurons present in the peripheral nervous system, which is represented here in yellow. So this octopus does not have a rigid skeleton, but how can he move? He can move using the isovolumetric constraints. So his muscles have this always the same volume, and he can make movements using this. And how can he control those movements? That's what we want to know. So previous studies on the octopus led to prototypes in robotics. So those are 2 prototypes created by the Italian Institute of Technology. And those are flexible octopus arm or flexible full octopus robots. They try to recreate the infinite degree of freedom of the octopus arm, but they are not there yet. That's why we need to continue to investigate further the motor control of the octopus. So this thesis was part of a bigger project that aimed to decipher how the motor command information is incorporated at each level of the motor control hierarchy. So it's aimed to understand clearly how does the octopus can control all of its movements. So in my thesis, I focused on the muscles and the peripheral nervous system. So first, how are organized the muscles in the octopus? In the octopus arm, we got 3 different types of muscles. The transverse muscles, here in red, the longitudinal muscles, so on 4 groups, and on the side, the oblique muscles. They are all organized around the nerve cord, which is composed of 2 parts, the axonal tract, where all the axons from the higher command center are located, and a chain of ganglia, which is presented here with the little red, yellow points. And those ganglia are containing the cell bodies of the whole peripheral nervous system. So we want to see the link between the ganglia and the octopus muscles. And for that, we have 2 working hypotheses. The first one is the topographic map. So where the motor neurons are clustering, and there is a spatial correspondence between the muscles and the motor neurons they are innervated with. The other hypothesis is an identity map, where the motor neurons are dispersed in the whole ganglia. So in order to determine which hypothesis is the closer to the reality, we used this experimental procedure. So we injected in vivo dextran, which is a retrograde tracer. We injected it in the muscles of the octopus, and after some time of incubation, so 1 week, 3 days, or 4 hours of incubation, we collected the samples and treated them for immunohistochemistry. Then we acquired images with a confocal microscope. With a confocal microscope, we can obtain images in the whole thickness of the sample. So we have image stacks for the whole sample that allows the 3D reconstruction of the samples. I was directly involved in the image analysis, as it was a completely remote thesis, and I had to reach 3 goals. So evaluate the injection methodology, so evaluate which incubation time was the most appropriate for our experiment, establish the image analysis method, and then evaluate the distribution, the numerosity, and the size of the dextran positive cells. All of this to answer this question: are motor neural cells topographically organized within the arm ganglia? So I will first talk about the evaluation of the injection methodology. So after image acquisition, we have those types of images. So here it is a whole sample image where you can see here the injection sites, and in the center, the ganglia. This image is a zoomed in image of the cortical part of the ganglia, so where all the cell bodies are located, and the different colors are representing different parts of the cells. So the blue is representing the cell nuclei. The green is representing the cell body, as it is acetylated tubulin staining. And the red, I don't think you see it very well here, but we've got red spots everywhere in the image, which is supposed to be the dextran staining. So here it's spots of red fluorescence in the whole sample. After 3 days of incubation, we have the same result as 1 week incubation. So little red dots everywhere in the sample. So the red fluorescence is in granular form, but we have different results with the 4-hour incubation. Here you can see that the dextran staining is only on a small fraction of the cells, and it's staining the whole cell body, and is not in granular form as previously. You can see maybe better here on this image. It's also 4 hours of incubation, and we have the same results at the previous image. So a few cells that are really stained in the whole cell body with dextran. So to conclude on those images and on the injection method, the 1 week and 3 days of incubation are too long, as the fluorescent signal was visible in a granular form. That can be due to the potential degradation of the dextran in the cell in lysosome-like vesicles, or because of the very low signal. We had a very low signal coming from the sample, so we had to increase the gain of the confocal microscope. That increased a lot the background of the image. So the 4-hour incubation gave good staining and so exploitable images. In order to exploit the images, I had to establish the image analysis method. So I used the ImageJ software, which is an open source software. I tested different plugins in order to make a 3D reconstruction, to count the positive cells, and also to measure the soma of the cells. So I compared automatic counting method and manual counting in order to find the most reliable way to analyze those images. So first, for the 3D reconstruction, I used the 3D viewer plugin, which gave a really accurate reconstruction of the image of the sample. So here on the right of the screen, you should have seen the sample in 3D, but as it is just an image, you see it in front. Maybe we will be able to show it after. I don't know. So give an accurate reconstruction of the samples. The 3D object counter and the colocalization object counter were used to count the cells. So here, it was supposed to be a video showing the method, but just the image also. So I used only one channel, so only the nuclei staining for the 3D object counter, because it's able to count only one type of fluorescence at a time. The colocalization can do on two types of fluorescence at the same time. So, for example, the nuclei staining and the dextran staining, but it gave the same results as the 3D object counter. So I put them together. Then the stack particle analyzer, which is another cell counter plugin, an automatic plugin that will count all the particles in the images. So in the image stack, it will count all the particles of each image, and there will be double counting of the cells with this plugin. But which was not the case with the previous plugins, as they were comparing the two adjacent image in order to see if the object was present in 3D in the 3D dimensions. Then I compare them to the manual cell counting, so selecting the positive cells manually. And I used a plugin, which is the cell counter, that just keeps tracks of the cell I counted with a little marker. And this marker is visible in all the images of the stack. So I can go through the whole stack and select the positive cells without double counting the cells. As a result, here is the difference between the automatic cell counting and the manual counting for the same sample. So as you can see, it's quite different, and that's because the image has a lot of background, and the plugins are quite sensitive. So they are too sensitive for our images. So we use the manual counting for selecting the positive cells for the other analysis. Concerning the soma size measurement, I did the same thing. So I used an automatic plugin to try to measure the soma of the cells that gave this image, which is not exploitable. So I did it manually using the polygon selection and the measure tool. So let's go for the third goal, the evaluation of the distribution, numerosity, and size of the dextran positive cells. So here on this table, you can see that there is a lateralization of the positive cells in the sample following the position of the injection site. Sorry. Okay. So after measuring the soma of the cells, we measure the soma of all the cells and the positive cells to compare them to see if there is different clusters of size. And what we saw in the first graph is that the distribution of the size of the soma sizes of all the cells and the positive cell is different. That was confirmed statistically. So the motoneurons stained with dextran are significantly larger than the other ganglia cells. We found also that there is a sandwich-like configuration of the dextran positive cells, so of the motoneurons. They are clustered together between two layers of smaller cells, as you can see on this image. So you have in red the dextran staining and in blue the nuclei staining. So you can see that there is a lot of smaller cells here and here. And in the middle, the dextran positive cells. So the motoneurons we are looking for. To come back on all the results, we have set up a good method for retrograde labeling the motoneurons with the dextran staining with an incubation of 4 hours. The 3D reconstruction of the imaging software gave an accurate reconstruction of the samples. And we found out that the best way to analyze our images is to do it manually, either for the cell counting or for the soma measurements. we identified two cell size clusters, so big cells that are the motor neurons and small cells that are other neurons. And those bigger cells are localized in sandwich-like configuration in between two layers of small cells. So going back to the working hypothesis, that gives the first indication of the innervation of specific muscle cluster by the motor neurons within the arm ganglia. And this is going towards the topographic map hypothesis, so that we have clusters of neurons inside the ganglia that are spatially correspondent to the muscle they innervate in the arm. That led to another hypothesis, which is that the motor command neurons coming from the higher centers, such as the brain, might reach and branch over a specific ganglia area where the motor neurons cluster in order to control the muscles. So to go further, what can we do? We can test other counting method to maybe find an automatic way to count the positive cells or measure the soma sizes. Or we can change the experimental procedure and do double injections of dextran, but with different fluorophores. Those double injections can be done in different parts of the same muscle types. For example, in the longitudinal muscle on the right of the arm and the longitudinal on the left, or in different muscle types, for example, in longitudinal muscle and transverse muscles. So that was everything for this thesis. I want to thank all the granting agencies and to thank a lot the Italian Institute of Technology members, especially Letizia Zullo and Alessio Di Clemente, for allowing me to work with them and for helping me a lot through this thesis. And also thank you to the MSc MARRES for making this thesis possible. Thank you for your attention.
Thank you. Right on time. So we'll start the questions with Elena.
Okay, so thank you for the presentation. It was really nice. You said that you want to try, new methods to count the cells. So what are you thinking of? And when you say manually, since you have done your internship by Raymond, you check just the image. You didn't have the possibility to check by microscope, right?
Yeah, that's right. I didn't check by microscope the images. Actually, that's Alessio, which is a PhD student, that sent me the images he acquired with the microscope. And I was using those images to analyze them and to find the good method to analyze them. So when I'm talking about manual counting, it's really manually selecting the positive cells or manually measuring the soma sizes using the manual tools from the ImageShift software, as I did during my thesis. And finding other methods will be to try to find other plugins that can make it automatic for those counting or measurements. As ImageShift is an open source software, there is hundreds and hundreds of different plugins that can be tested. And I didn't test all of them, so that can be done. And maybe we can find some plugin that can be used for those kind of images.
Okay.
Elena. Stefano?
Yes, thanks, Hugo, for the presentation. It's a pity. I would have liked to see your 3D videos. But
okay, you're going to see them.
You will have the opportunity another time. So you were talking about when you were the first point, so evaluating the injection methodology, you were talking more about, you presented more about the time, but what about the space, the place of injections? Because I suppose that you want to inject closer to at the beginning of the arm, or how is it choose the position rather than of injection? I know you didn't do it practically, but
Yeah, it was in the median part of the arm, so more or less in the middle of the arm that was selected. And the samples were really small. There was a thickness of around 40 microns for each sample. So that was directly on the injection sites.
Okay, so the position of the injection site didn't change. What it was changing was the time of the incubation, the detections, and then as a control for what did you use or as a experimental control? So do you have some morphological data on the sections that you have been looking for your fluorescent? So do you have something of I don't know, hematoxylin eosines or electron microscopy?
Maybe you can see here the 3D reconstruction.
Yeah, a little bit. Yeah.
The second you will see it entirely.
Thanks.
Yes, we have it, I think. Cool. She can comment it.
Yeah, so in the 3D reconstruction, there is so the three colors we saw in the other images. So in blue, that's the nuclei staining. In red is the dextran staining, which you can see here clearly the granular form. And in green, it's the acetylated tubulin staining, which did not really penetrate in the sample for this sample. That's why we got just not the whole image in green.
Do you want to show another video?
Yeah, I have a supplementary slide, which is written no dextran injection.
You can just tell me on the left.
Yeah, it's slide 33, 4, maybe more.
This one, 37?
No, there's supposed to be others.
There is no more.
Yeah. Well, there was supposed to be another image with no dextran staining, so the same sample treatments, but with no injection of dextran. And on those images, there was a lot of autofluorescence in red of the ganglia cells. That's why the 1 week and 3 day incubation time were not good enough to be used for the analysis, because we could not distinguish between the autofluorescence and the dextran staining.
So for your counting, did you use a section from more than one individual or different section from the same arm and the same individual? So the number is the individuals, but also the number of arms.
We used different arms from different individuals. So each sample is from one arm from one individual. We did not use several samples from one individual. And going back to the counting of the positive cells on the graph comparing the methods, that's only for the same sample. I counted with the different methods one sample, and that gave those results.
Okay, and one of the justification of the huge difference between the what it's counted by the plugin of ImageJ and what you counted manually. Do you think there is a room for improvement from the automatic size? Do you think you with the same plugin, or would you use a different plugin to have a less sensitivity, or changing? I suppose you already changed the color in a black and white before the quantification. So do you have something to comment about that?
Yeah, so actually before using the plugins, I treated the images, so putting them in black and white, adjusting brightness and contrast, and putting a threshold on each image to have the most accurate analysis. But all those plugins are mostly particle analyzer. So when there is one pixel or a little group of pixels that is colored and not and over the threshold, it will be counted as one cell. That can be adjusted with in some plugins. That's what I try to do. That's why some plugins have a closer number of cells counted to what I counted. But so there is still room for improvement for those plugins, but also we can try to develop other plugins that can be more like the colocalization plugin. So where we can have select only the cells that are positive to nuclei staining and dextran injection, for example, and have so a possibility to set a minimum size a minimum pixel size to make it positive. In most of those plugins, there was no minimum size or circularity of the, let's say, of the positive color they measured. So that's why we got those high numbers. When the plugin is counting, it's just looking at if there is color or not. And we can adjust that by saying you need to see if there is color or not, and if this color is on this size of pixels. So this area is colored, so it can be considered as a positive cell. And this area is too small, so it's not considered as a positive cells. Okay. That can be improved.
And just sorry, yeah, pardon. Yeah,
Stephanie, I'm sorry. Yeah, maybe a really last, really.
Yeah, you're right, but it's
okay.
Yeah.
Thank you, Stephanie.
Sorry about it.
We have to proceed. Maybe Robin.
Okay, so I'm gonna stay a bit on the same topic about the image processing. So you only had 223 cells, if I heard. So do you think there's actually, in your case, for like dealing with the same data, is there any advantage of trying to automatize the image processes? And if you forget about imaging and plugins and stuff, what would be another way to try to automatize this detection process?
Okay, so the advantage of automatization of this process is we can increase the number of samples treated in a short amount of time. That will increase the number of results. That will be so we will be able to strengthen our conclusions, our biological conclusions, and in a way to automatize those kind of analysis. It might be possible to do it with some machine learning in order to have a program to count the positive cells.
Yes, machine learning is a broad term, but yeah, in this case, like deep learning to identify this kind of cells when there's very few, it's probably going to be relatively easy and efficient. And I know it's like widely used in medical. For medical research. So, yeah, that's it. Thank you.
Oh, okay, thank you, Anne?
Yes, thank you for your presentation. So I just had one question. How do you recognize a differentiated motor neuron with your images? I'm just interested.
So in the images,
okay, I'm back.
So much? Okay, just a second. So in the 4 incubation images, we can see clearly that the images are stained with dextran. Just here. So you can see in red all the circles, circle-like red fluorescence is a positive cells, positive cell, and those are, sorry, can you repeat your question?
Yes. How can you differentiate, because it's in your abstract too, a differentiated motor neuron?
Yeah,
from others.
We differentiated them from the other ganglia cells. So that the difference is mostly the soma size. So the cells stained were larger than the other cells, and that's the main difference between the motor neurons and the other ganglia cells.
Okay, thank you.
Thank you. And Laetitia?
I think no, Laetitia.
No, Laetitia, I'm sorry. Yeah, I'm just sleeping. Sorry, Laetitia, it was just saying hi. I'm sorry. We're coming back to you after. Sorry. So let's move to Aldine.
Hi, Hugo. So thank you for your presentation. I have a question actually related to the one from Anne, because so at the beginning, you mentioned that with the dextran, you got granular staining, and it was not what you were expected in this experiment. So it made the analysis a little bit more tricky, but the dextran is lipophilic. So you will wait for it to be like a dotted or vesicle somehow in the arm of the octopus. So can you comment on that? And the second thing is that how do you make the link between dextran and motor neuron?
Okay, so the dextran is lipophilic, so it will have this granular form, but with a low incubation time, so between the injection and the sample collection, it's less grouped in vesicles than with the longer one. So in the forward incubation, the dextran positive cells are let's say a lot of really tiny dots of dextran. As it is lipophilic, it's in a really little granules diffused in the whole cell body. And with more incubation time, they are all grouped in, more or less one vesicle per cell. But this is not distinguishable between the dextran staining and the autofluorescence of the sample. That's the reason we use only the forward.
Okay. And go ahead.
Yeah, the link between dextran and the motor neurons. So we used dextran, we injected it in the muscles, and it traveled along the axon of the muscle of the motor neurons, innervating the area injected, and yeah, that's how it got to the soma of the motor neurons.
Okay, but how people previously highlighted these motor neurons in the arm of the octopus? Is there any study that really show I don't know, specific labeling in these motor neurons, or it's just based on the morphology of those cells that you can recognize them?
It's based on it can be based on the morphology of the cell and also on the location, because in the ganglia there is neurons. Yeah, that's I think there is only neurons in the ganglia. So yeah. I'm not really certain of this, but there is nearly only neurons in the ganglia, so that's how we can say that those are neurons.
There are neurons, but in the ganglia there is motor neurons and other.
And,
no?
Yeah. So this sorry. So we can say there are motor neurons because they are linked directly to the muscle. And it's because of their organization, and they're linked to the muscle.
Okay, okay, thank you.
Thank you, Paula.
I have a question about the motoneurons, and I'm just wondering if they're only linked to the mechanical movement. There could be other motoneurons in cephalopods that they can have other roles.
That can be possible, but here in this study, we're focusing only on the muscle movements. So here in the arm, only the arm muscle movements, and those motoneurons that are staying are linked to the muscle in order to make them move. Yeah.
Okay,
yeah.
I say the reverse. How are you, but it's some way very similar to the question that you had before. It's how do you know that you really have the really stay neurons linked to the muscles that you want? And the other question is how were these little the little cells that you can have distinguished all around the motoneurons?
Okay.
I don't know if there is a link between two questions. This is why I asked you.
Yeah. Okay. So those are motoneurons that were staining because first the dextran is not passing through the cell membrane. So when injecting in the muscle, we are actually injecting the dextran in the terminal part of the axons that are innervating the muscles. So we are sure that the stained neurons are linked directly to the muscle, so they are motoneurons.
Okay.
And the other cells that are present in the ganglia are potentially sensory neurons, but we didn't identify them, so we are not sure about what kind of cell they are. They are neurons, but we don't know if there is sensory neurons or not.
Okay, and you never saw any labeling on more on the surface, more on the epiderms, link more to the chromatophores in cephalopods? Because there is this movement of the chromatophores linked to the motoneurons in cephalopods, but not with this method.
Not with this, as it is in an injection in the muscle.
In the muscle, and this is what I didn't catch. Okay, thank you.
Okay, yes, please.
Yes, thank you for the presentation. And before asking my question, I'd like to congratulate for your work because doing an internship remotely without touching your material, and that you've been presenting it, saying we when you talk about injections. So I think you did a good job in imagining to be there and just being in front of photos, which must have been very prestigious. So my question is related to your graph with the different measurement, and I would like to know how much confidence you have in your measurement as manual measurement, and what type of variability there was between those counting. Are those methods, the other method might be different to the manual counting, but maybe more reproducible over time. So do you have any comment on that?
So,
because I don't think there were standard deviation.
No, there was no, because it was only for one sample.
I see.
So that was one try for each to see the difference for one sample of the difference of the method. But concerning the confidence for the manual counting, so the counting I did, so the counting I did was also did for some sample, not all, by Allazio. We checked and we had the same results. So we're two different observers to have the same results. So we have quite a good confidence about those numbers. And concerning the automatic counting, those are the numbers for one set threshold and one set brightness and contrast. So when you modify the threshold of the image, we can have really different different numbers from the same plugin. That was really the complex part of those plugins is, for example, the stock particle analyzer for this sample had 36,000 cells. But if I modify the threshold a bit, like a bit more or a bit less, it can vary from several thousands. So this plugin was really sensitive. The other was less sensitive, but there was still this difference. But if we set the exact same threshold for each sample, that can be reproducible.
Okay. Because I was looking at, of course, the other one is, I don't know how many order of magnitude difference, but the 3D object, I mean, 446 and 223, maybe, I was not sure on how much, yes, variability, but if it's sensitive to other parameters, continue to count manually. Sorry. Thank you.
Thank you. So that's all. Thank you, Hugo. Did I forget someone?
Eric, peut-être?
Eric? I forgot Eric.
Yes, me. Hey, Hugo. Thanks for your presentation. Very interesting. I have a question that sort of is based on your future perspectives, and that put everything, all the questions that you had back in context. So you propose that it would be interesting to inject 2 different DEX strands. It means that everything will be repeated. So what would you recommend now that you know the sort of the limits of the analysis and what you can do, what you can't do? What would you recommend to do from A to the end to try to optimize the whole pipeline?
Okay, so in order to optimize the pipeline for further experiments, yeah, what we can do is mostly doing it manually. The staining of the different fluorophores can be done easily as an experimental procedure, and that's really the complex part here is the image analysis, and that's the process we need to optimize. So that's for now the most reliable method is the manual counting. So yeah, I don't see really how we can optimize it for now.
Okay, let's say I'm asking this question because at one point you mentioned that it was tricky to distinguish between a particle and autofluorescence. So there are some imaging acquisition tools, equipments, spectra confocal, for example, where you can sort of eliminate all the autofluorescence at the beginning. You have some standardization to do at the beginning, but then once you have this, you don't have to deal with any autofluorescence anymore, once you know what the spectrum of the autofluorescence is. And this was a little bit towards this direction. If at the IIT they have access to it, if that would be something interesting to do, because once you do that, all the plugins you use might be used for the machine learning, because if you have a machine learning algorithm, you still have to deal with the autofluorescence that may hamper the thing. So I was going towards this direction. What can you imagine in trying to optimize the workflow? Because you were trying to use different plugins. It's an endless story. Try it, but if there's something else you can do, it might help.
Yeah, so yeah, as you said, having a confocal that can solve the autofluorescence can be one of the most useful optimization of this process to have to avoid the autofluorescence to be present in the samples, but also the incubation time was set at 4, where we can distinguish easily the autofluorescence from the dextran staining. So that solved this problem already. Yeah.
Okay. Thanks a lot.
Thank you, Hugo. I think I forgot more people. I'm sorry for that. Oui, okay. I think I have to drink some water.
An ecological question. It's about how is this nervous system an advantage in its living environment?
This is an advantage because the octopus is able to control each part of its arm, and as it is very flexible, it can do a variety of movements, and that helps him a lot for example, going through small spaces. and also catching prey. But yeah, the fact that it can do really precise movement with this very flexible arm is really interesting for him, for it as a hunter, because he can go with its arm in little cavities where it cannot go completely and seek some prey, for example, so little prey. So that's really an advantage for him to be really flexible and be able to control each part of its body like this.
It can compete with the other vertebrates.
Yeah.
Okay, thank you. Sorry. I think I have to take a glass of water, maybe also a coffee, and we will be back in 10 minutes. But thank you, Hugo. Congratulations and good luck, of course. So if you're following us on the YouTube, after a glass of water and a coffee, we will be back with Juliette Armeni, but this presentation will be private. Juliette will talk about optimization of fucoxanthin production with Tisochrysis lutea, so it will not be streamed live. And then we will talk about quantifying coastal fisheries enhancements in Portugal, close to the Rio Formoso lagoon, with Zineb Sada. So we will come back with Zineb somewhere around 4:50, between 4:50 and 5. Voilà. Toi aussi, tu reprends un petit verre d'eau.
Merci, Hugo.
Ah.